Hi guys and welcome. This is a HL video on data analysis questions. So looking at some of the skills that you specifically need to develop in order to be able to answer these kind of questions. I've got to say that they prove to be some of the most challenging type of questions that students encounter in IB, but also some of the most gratifying because they really show you the development of your skills when handling data, and really so much use as well when you do your IB biology IA, okay? So your ability to analyze data, to evaluate data is really important. So we're going to go through these questions now. What I want you to do is, whilst the video is playing, I want you to, and I come to a question, I read out the question, I want you to look at it very carefully, pause the video and try and answer it. Just on paper, just write your answer down somewhere, and then we'll go through that answer together and kind of pick out what you needed to have done. So it's kind of an activity for you as well. So it's not just you sitting there listening. All right, so because we're looking at genetic modification at the moment in biology, I thought it would be important that the type of question we go through should be one on GM foods. So let's look at this. There's going to be some terminology. There's always terminology that you come across in these questions that you won't have encountered before. And so you've got to be quite adaptable. You've got to sort of approach it with an open mind, thinking that the bigger concepts you just don't know the lower down specifics. So don't get too kind of wigged out by it all. All right, so let's read through the question first of all. The question will always have a question stem, so the initial giving you kind of the background to it. It will then give you a set of data which you're going to have to analyze. Further throughout the question, there'll be more data as well that you kind of have to look at. Anyway, gibberellin promotes both seed germination and plant growth. Researchers hypothesized that the gene GID1 in rice, Oryza sativa, codes for the production of a cell receptor for gibberellin. The mutant variety of GID1-1 for that gene leads to rice plants with a severe dwarf phenotype and infertile flowers when homozygous recessive. It is suspected that homozygous recessive GID1-1 plants fail to degrade the protein SLR1, which when present will inhibit the action of gibberellin. So the graphs show the action of gibberellin on the leaves and alpha amylase activity of wild type rice plants, which is WT, and their GID1-1 mutant cousins. Okay, so a lot of kind of terminology within that, a lot of information. Okay, you've really got to try and interpret the language. So first of all, what it's telling is that plants that have the gene GID1-1, okay, this mutant gene, are going to have a dwarf phenotype and infertile flowers if they're homozygous recessive for GID1-1. Okay, if they are the wild type of gene where they haven't got this mutant gene, they're going to grow normally. Okay, also by looking at the data here, you can see that if they've got the GID1-1 gene, the alpha amylase activity is activated, or a high level of activation under different concentrations of gibberellin. And for GID1-1, it's not activated. The amylase is not activated at all. Okay. So the first question is, state which variety of rice fails to respond to gibberellin treatment. Okay, so looking at both graphs, you should be able to see that both GID1-1 rice, okay, both of them, okay, fail to respond to gibberellin treatment. Okay, so the answer for that is, of course, is GID1-1. Now, what you're going to be very careful with these questions is these key words here. So in this one, it says state. We call that word at the start of the question a command term, and it has a specific definition which you need to learn. So state simply means give a specific name, value, or brief answer without explanation or calculation. So it's a simple one-word answer, one-letter answer, and of course the answer you're looking for is GID1-1. Okay? The next question we come to. The activity of alpha amylase was tested at successive concentrations of gibberellin. Determine the increment in gibberellin concentration that produces the greatest change in alpha amylase activity in wild type rice plants. So again, just before you pause this video, figure out what the command term is. And you can see it here, it's determine. Now the word determine means find the only possible answer, okay? So there's only one possible answer for this. So let's go back to our data. It's the activity, so we're looking at the alpha amylase graph, and we want the increment in gibberellin that produces the greatest change in the amylase concentration. So in terms of a graph, of course, we're looking for the greatest period of change, or the increment where there's the greatest period of change in this graph. And so obviously that's the steepest part of the graph. So that's going to be between here and here, okay? So in terms of the increment that we're looking at, it's between 10 to the minus 7 and 10 to the minus 8 moles per decimeter cubed. So the increment, remember, is not one specific value, it's the change. So from one step to the next step, okay? Right, next question. Outline the role of alpha amylase during the germination of seeds. Now again, that word outline, give a brief account or summary of the role of amylase during the germination of seeds. Okay, so this might be something hopefully you might remember from GCSE. But the role of alpha amylase during the germination of seeds is it's going to start to break down the starch that's stored in the seed, okay, and it's going to change that starch into maltose. That maltose can then be used for respiration to generate ATP, which is used then for the growth of the plant and other metabolic processes. So really, really important thing. Okay, here we approach our first high-level question from this question. It says, discuss the consequence of crossing GID1-1 heterozygous rice plants amongst themselves for food production. Okay, so this word discuss, what does that mean? So when we're talking about our command terms, discuss means give an account, including, where possible, a range of arguments for and against the relative importance of various factors or comparisons of alternative hypotheses. So it's quite a tricky question. You can see it's worth 3 marks, okay? So again, pause the video and now try and answer that question. Okay, so you've been through that question and you've looked at the thing and hopefully you've come out with the right answer. So obviously, if you're crossing GID1-1 heterozygous rice plants, we should remember, if we think about it in terms of a Punnett square, that 1 in 4 of those plants is going to produce seeds that are homozygous recessive. Okay, and we know that those homozygous recessive plants, that 1 in 4, are going to, by looking at this information here, they're going to have severe dwarfism phenotype, and they're going to have infertile flowers. Okay, so what that means is with dwarfism that they're going to get less growth because they're going to be dwarf plants. They're not going to be responsive to gibberellin. They're going to be these ones down here. The flowers are going to be infertile. Okay, so an infertile flower cannot produce seeds. And so ultimately that's going to lower the rice production. Again, okay, so it's again linking all those ideas together into one answer. So it's a 3-mark question. Any of those 3 points that I've just discussed there could be used in your answer. Okay, so now we're moving on now to look at our second bit of data. Okay, again, it's going to switch it up on you nice and quickly so you never really get a full handle on things. So it says most rice varieties are intolerant to sustained submergence underwater and will usually die within a week. Researchers have hypothesized that the capacity to survive when submerged is related to the presence of 3 genes very close to each other on rice chromosome number 9. These genes were named sub1A, sub1B, and sub1C. The photograph below shows part of a gel, and it shows the relative amounts of messenger RNA produced from these 3 genes by the submergence intolerant variety, O. sativa japonica, that's these guys, and by the submergence tolerant variety, O. sativa indica, that's these guys over here, at different times of a submergence period followed by a period of recovery out of water. So you've got, here's the 3 genes, okay? Each of these shows how much RNA is being produced of those particular genes. Remember, RNA relates to particular genes. And it shows how much of it's being produced on different days of submergence and in days of recovery, okay? So it says, first of all, determine which gene produced the most messenger RNA on the first day of the submergence period of variety O. sativa japonica. So how are we going to do that? So again, that word determine. Determine means find the only possible answer. So we're looking for a very simple answer. We're looking at O. sativa japonica, which is this one. So, and we're looking at the first day of submergence. So which of these 1, 2, 3 genes was expressed the most on those 3 genes? Well, pause the video and have your best guess. And unpause now, and hopefully you've realized that sub1C was expressed was expressed more than the other 2 genes on day 1. So the answer, of course, is sub1C. Outline the difference in messenger RNA production for the 3 genes during the submergence period for variety O. sativa indica. So it's switching it up on us now. We're going over to the other data set, which is here. And this command term outline simply means give a brief account or summary. So nothing too detailed. So when you're answering this question, you want to make sure that, just hold on a second. All right, it's a nice simplified answer. You want to make sure that you're using the right data set. So you're looking at O. sativa indica, and that you look at each gene one at a time. Okay, so sub1A, sub1B, sub1C. So let's start with sub1A. So you can see that sub1A is expressed strongly or the most and produces the most RNA given by the whitest color throughout the submergence period. Sub1B always has the lowest expression, or it produced the least messenger RNA, denoted by the least coloration. And sub1A is expressed sub1A is expressed produces messenger RNA for the least period of time, but quite strongly on days 3 onto days 7. Okay? All right. And then it says, using only this data, deduce which gene confers submersion resistance to rice plants. So which of the 3 genes can be used to confer that submersion resistance to rice plants? So when it says deduce, this is our buzz term. Deduce, again, is quite a complicated idea. It means reach a conclusion from the information given. Reach a conclusion from the information that you're given. So what information do we have? Okay. Well, we know that indica is the variety that is showing submersion resistance. Okay. So this one here has the submersion resistance. Okay. Japonica does not show submersion resistance. Out of the 3 genes that's here, there is 1 gene which is only expressed in sativa indica, and that is sub1A. So it can be deduced from that information that sub1A is the gene that gives us gives submersion tolerance. Okay? Anyway, I'm going to finish this video here and I'll finish it off shortly. Thank you for watching.